Okay, I'll give folks a few more seconds here to wrap up with the quiz. So if you're just joining us, please go ahead and fill out the quiz. There should
Where
be.
is the quiz?
Uh, Sometimes you can't see it if you've got pop-up blockers and things like that. It depends on what you're doing, but it should just show up as a window. All right, we've got 16 of 28 that have done it. We'll go ahead and revisit the quiz too at the end. So I'm just going to go ahead and end it.
I'm not seeing it.
It's
Yeah, I'm
okay.
not, I'm not seeing it either. Um,
That's all right. I think, like I mentioned, it depending on what browser you're using and like the settings on your browsers, you might not see the quizzes pop up. So all right. With that, I just wanted to pass it off here for the to Dr. Braham Dhillon. He's the state pathologist, plant pathologist that focuses on palm diseases, and he's out of Fort Lauderdale Research and Education Center. And we're very thankful to have him here today. So with that, I'm going to pass it off to Dr. Dhillon.
All right. Thanks, John. Hello, everyone. Good afternoon. I cannot see anyone, but so I'm just going to talk to the screen here. I'll try to keep it simple and straightforward. Again, my name is Braham Dhillon, Assistant Professor of Plant Pathology. I'm based in Davie at Fort Lauderdale REC. I'll be talking about fungal diseases of palms. That's the primary mandate of my position. I'm 70% research, 30% extension. I'm trying to work on other tropical crops and other diseases, but fungal disease of palms is what I specialize in. Just before we get started, the first few slides are about plant pathology in general. So when we say disease, it results from a dynamic, detrimental relationship between an organism that parasitizes, so the organism that is causing the disease is the pathogen, and the organism that is being affected is the plant host, is the host. It could be a plant or it could be a mammal. In the broadest sense, anything that can negatively impact the host is disease. But in the narrow sense, disease is caused by living organisms. On the left side, or is a list of organisms, that includes viruses, bacteria, fungi, parasitic plants, nematodes, and insects, mites. They cause disease. And these are the biotic factors or the living factors. The abiotic factors, or the non-living factors, when they negatively impact plant, they are characterized as disorders. So there's a clear distinction between a disease and a disorder. And there's a third category of injury where heat or frost or mechanical damage or herbivores when they feed on plants, that's injury. But disease and disorder can be differentiated based on living versus non-living organisms that are negatively impacting the health of the plant. So the study of plant diseases is plant pathology or phytopathology. It comes from the word pathos, meaning suffering, and logos is study. Phyto here is the plant. Again, disease is any abnormal condition that alters the appearance or function of a plant. And the key components of a disease are, there is an organism that is negatively impacting the function of a plant. And the impact is there is a reduction in quality or the quantity of the harvested product. Not in the case of palms per se, you're affecting the quality of the product, but in terms of quantity, you can think about, fruit crops or row crops, you're decreasing the yield. Diseases can decrease the yield. And it is a process that changes or progresses over time. It is not stagnant. You can see, when disease appears, it's not instantaneous. There are lesions that start developing and then they progress over time and eventually, in some cases, can kill the plant. The history of plant pathology is as old as the history of agriculture itself. Romans had a god of rust called Robigus. Robigelia was a festival that was held every year on April 25th. Basically, coming from the rust, a red dog was sacrificed. and during the wheat ripening period, when the Sirius star was visible in the skies. There are different schools of thought on what rust could be. Rust could also be, their weapons were mostly iron, and so rust could also be the, rust that was affecting their weapons. But as a plant pathologist, I believe, or my train of thought is that, they were, the Roman god of rust was related to the rust disease that happened on wheat. So in terms of plant pathogens that cause disease, I listed of 6 different categories. Here I'm showing examples of pathogens. In panel A and panel B, you can see a close-up of, in panel B, you see all these nodules or modules that look like they're fungal spores on branches of fungal hyphae. In panel A is a petri dish with media in it and with a single spot that grew out radially. And so a lot of these pathogens, fungal pathogens, we can actually isolate from the samples you submit to the lab, and we can grow them in pure cultures, and we can easily work with them. And that being said, not all fungi can be cultured in the lab. Like I mentioned, rust that comes on a lot of different crops. These are obligate pathogens. They cannot survive without their host. But other pathogens that can survive with the host, we need to give them carbon and nitrogen and other micronutrients. They can easily grow out in the plates, and we can manipulate them easily and study them in the lab. The panel C here, you can see the small cells here in the middle. Hope you can see the mouse. The base structure is a stomata. This is the stomatal opening, and the tiny cells you see are bacterial cells. And based on the magnification, you can see the higher this number, the smaller the organism is. So fungi are larger, in general, are larger than bacterial spores. And in the deep panel here on the bottom right corner with the 5000x magnification, the cells that you see are phytoplasmas. Phytoplasmas cause disease on palms as well, including a number of other crops. Lethal yellowing, lethal bronzing are caused by phytoplasma. These are very closely related to bacteria. The major difference being they don't have a bacterial cell wall like the bacteria have. And these are, you can see with the magnification, you need a higher mag to see phytoplasma in the plants. And these are obligate pathogens, so they cannot survive in the media. They need a live host. So it's harder to study phytoplasmas as compared to, say, bacteria or fungi that can be cultured in the lab. Other classes of pathogens include, there are plants that are parasitic on other plants. So parasitism comes from the fact that they cannot manufacture their own food. Plants are what we call as autotrophs. They can harvest sunlight and convert that into food or energy. And other organisms that cannot convert or cannot produce their own food are heterotrophs. So fungi are heterotrophs. They depend on other organisms for their food. And these parasitic plants, they lack the mechanisms to make their own food, so they depend on other plants for their food. That's panel A in this slide. And these strands that you see are parasitic plants. The other category of organisms are viruses. Here you can see these hexagonal cells. These are actually virus particles. And different viruses come in different shapes. The coronavirus looked like the name comes from the virus looks like a crown. That's the name corona. And here, these are hexagon virus particles, and based on the mag, you can see 200,000x magnification is needed. So these particles are really, really small. And in the bottom panel, C and D, these are nematodes. These are parasitic worms, usually infect roots. There have been cases where nematodes can cause disease on the whole plant, but normally they infect roots and they can cause, galls or other malformations on the root and affect how plants can absorb nutrients and eventually kill the plants. So these spindle-shaped objects or these long rod-shaped, needle-shaped, objects here, these are nematodes, and these are fairly large. Some of them are even visible to the naked eye. So plant pathogens come in different categories. Today, we'll be just talking about fungal pathogens. When we talk about diseases, there are 2 important terms. when you're describing a disease to someone, the signs are the physical evidence of a pathogen. For example, if you see a mushroom fruiting body, if you see smaller fruiting bodies that appear as pinhead objects, I'll talk about those later. Or when you're dealing with plant samples in the lab, you can see actually, bacterial ooze coming out of the disease samples. Wherever you can see a physical evidence of the pathogen, that is a sign, as opposed to symptoms, which are visible effects of disease on the plant. Any detectable change in color, shape, function of the plant in response to the pathogen or a disease-causing agent is a symptom. Lesions that you see on a plant that change over time, that is a symptom. And if you, for example, in Ganoderma, that I'll talk about in a little bit, when you see the fruiting bodies on the palm trunk, those are, actually signs of the disease. This terminology, having this terminology helps you talk have similar terms that can help explain the symptoms and signs of the disease and can give you a common terminology to discuss the disease in a more effective manner. So disease does not happen or instantaneously. There are things that need to come together to cause disease. So there are at least 3 aspects to disease. You need an organism that has the ability to cause the disease, that is a pathogen. Not all fungi can cause disease, so there are non-pathogenic organisms. So if that is present, disease won't develop. If you have a host which is resistant, there will be no disease. You need a host that is susceptible. You need favorable environment. You need the right conditions, right temperature, right moisture, right soil conditions, soil pH, soil temperature, that is favorable for the disease to happen. We see more samples coming into the diagnostic clinic during the rainy season. That's when you have cloudy skies, lower temperatures, a higher moisture content that can favor certain diseases. So these 3 components: a pathogen, an organism that has the ability to cause disease, is pathogenic. You need a host that is susceptible to the disease, and you need a favorable environment. These 3 come together, then only disease will happen. Otherwise, if one of these components is missing, disease cannot happen. In terms of how disease progresses, this is known as the disease cycle, as opposed to the fungal life cycle. Disease cycle is how the disease occurs in the environment. You have a dormant period. In temperate regions, it's the cooler environment when fungi cannot grow really that well, so they overwinter. In Florida, it is oversummering. During the summer months, it's really hot, and it's not really conducive for fungal growth, so that's the dormant period. But once the dormant period is over, the inoculum that is present will start developing, start producing primary inoculum, and then this inoculum is spread by different factors, by rain, by wind, can be carried by vectors like insects or other organisms, and can reach the host. And if the host is susceptible, these spores will germinate, penetrate the host and cause infection, and then you start seeing these symptoms develop from chlorosis to necrosis to eventually, in some cases, these lesions merge and take over the whole plant and kill the plant. Some of these diseases, they only have one cycle of the disease cycle during the conducive period. In other diseases, they can infect the plant and produce more spores and keep spreading from plant to plant as they produce more inoculum. So that is the secondary inoculum or dissemination of the pathogen. And when, as the season ends, for annual crops or for perennial crops like palms in the landscape, when the conditions become less favorable, these organisms want to survive. They produce overwintering structures or oversummering structures, and they stay dormant. And then this disease cycle carries, is reoccurring as environmental conditions change. Or so the reason for me to discuss this is, if we can understand more about the disease cycle of what stages occur during the disease, it's easier to disrupt the disease cycle, and that is our goal. If we can disrupt the disease cycle, we can either suppress the disease or stop the disease from occurring, or we can stop the disease from spreading. And that is the end goal of developing these, of studying these diseases and understanding the disease cycle so we can develop better disease management strategies. So that's all I'm going to talk about diseases, just a general background of different factors that can cause disease, what of the terminology that we use when we're talking about disease. So I'm going to switch gears and talk about POMS in general. All right, so I'm sure everybody is familiar with the anatomy, just so that everyone is the same page. Majority of the palms that we see in the landscape, they have a single trunk, and there is a single growing point, which is hidden somewhere in the top of the plant. That's the meristem. And if the trunk, since it's a single trunk and a single growing point, if these can be attacked or the disease can get to these points, then it is lethal. And I'll come back to that in the next slide. But in terms of palms, majority of these palms, you have these fronds, which collectively are known as the crown. Some of these palms, they have a green structure where the fronds are wrapped around the stem. That's the crown shaft. And the unopened leaf is called a spear leaf. And so, like I said, a lot of the palms in the landscape are single trunks and they have single growing points. There are very few palms that are branching or that have multiple trunks. And in terms of fronds, you have fronds that are pinnate, where you have individual leaflets attached to the central branch, which is called the petiole or the rachis. So petiole and rachis are used interchangeably. In the palmate, it looks like the palm of your hand. There are no leaflets. All the leaf is like a fan, and it's attached to the petiole. And there is an intermediate costa palmate, where it looks like it's an intermediate between pinnate and palmate. But these pinnate and palmate leaves are used to, characterize palms or broad classification of palms into two different categories. So coming to the diseases, broadly speaking, we can distinguish or characterize these diseases as lethal or non-lethal. So like I said, majority of the palms have single trunk or single growing point. If you can get to that, if the disease can get to that, the trunk or destroy the trunk or the bud, then it's lethal. Non-lethal diseases generally colonize leaves, but this demarcation is like, can be considered as a line in the sand. Some of these non-lethal diseases can become lethal if these are not effectively managed. The lethal diseases that I'll talk about today are Fusarium wilt, Ganoderma butt rot of palms, Thaliopsis trunk rot, and bud rot caused by Phytophthora, which is not really fungal, but I'll come back to that. And non-lethal diseases I'll talk about, petiole blight, Pachylotiopsis diseases. There are leaf spots caused by a lot of different organisms, and a false smut caused by Grapheola. So once again, non-lethal diseases can become lethal if they're not managed properly. So first one I talk about is the Fusarium wilt. It's caused by Fusarium oxysporum. Oxysporum is a unique pathogen. It has, normally when we discuss pathogens, we have a binomial name. You have the genus and the species name, Fusarium, oxysporum. In oxysporum, there are special categories or forma specialis that depend on which host they infect. So Fusarium oxysporum can infect a lot of different hosts, but something that infects, say, tomato, normally or will not infect other crops. They are really specific to tomato. Other Fusarium specialis, forma specialis of oxysporum that can infect watermelon will not affect tomato or palms. Similarly, oxysporum that infects palms cannot infect other hosts. So they're really specific to their host. And that's where the forma specialis concept comes from. It's the host specificity. In palms, so the Fusarium wilt, the symptoms they present as one-sided leaf death that you can see here. You can see a streak running along the petiole or the rachis, and one side of the fronds are dead. The leaflets are dead, the other is still healthy. This is the early symptom. As the symptoms progress, the whole frond will die out. And the symptoms, they progress generally from the lower part of the canopy and move up into the canopy towards the spear leaf. So the older fronds show these symptoms, and as they're progressing, they will move to the younger fronds. So the older frond will die out completely, but the younger frond will have this one-sided leaf death. So when you're looking at the canopy, you can look for these one-sided leaf death, and these are usually telltale sign for Fusarium wilt. And I say usually because I'll talk about another disease that can present one-sided leaf death as well. So in palms, there are two forma specialis of Fusarium oxysporum. One is canariensis, as the name suggests, it primarily infects Canary Island date palms. And the other forma specialis is palmatum, which infects Queen and Mexican fan palms. And morphologically speaking, you cannot really tell them, even if you can isolate them from the palm. Based on their morphology, we can tell them apart. We need to look at their genetic signature of these two organisms, or of the Fusarium oxysporum that we have isolated, and tell whether it's palmatum and canariensis. The reason that's important is, palmatum is really aggressive. It usually kills the palm, in about 2 months, whereas canariensis can linger on for a year or more. Again, some more symptoms here. Fusarium oxysporum or Fusarium wilt is actually what we describe as a vascular wilt. So as Fusarium grows, it's actually colonizing the xylem vessels, which conduct water. You can see here in the right panel here, you can see a browning of where the browning of the pedicle, you can see inside the tissue is brown. Here, the xylem vessels, the Fusarium spores are growing in, and they're occluding those vessels, and no water transport is happening. So you see this leaf death. And so the pathogen is actually not present in these leaflets, it's present in the petiole. So the best tissue to sample when you're looking at, one-sided leaf death or something that looks like Fusarium wilt is to sample the petiole. And it is sometimes challenging when you have such long, tall palms growing in the landscape. But the petiole is the best tissue to sample if you want to get an answer on whether Fusarium is causing those symptoms or not. So Fusarium wilt, like I said, caused by palmatum on queen palms and Mexican fan palms, or Washingtonia palms. It's really aggressive. It can kill the palm in about 2 months, and the palms die so quickly that these fronds really don't have enough rot or breakdown of the tissue that these fronds don't droop. So this is actually like a mummified queen palm. And so if you're sampling, you can see here the spear leaf, or the youngest open leaf, is still a little bit green. If you're sampling, I would suggest sample this petiole here, which is still in the process of dying and not completely dead. In terms of diagnosis, like I said, initial symptoms of the one-sided leaf death, they can be confused with petiole or rachis blight. I'll talk about that in a little bit. And that also shows up as a single or a one-sided leaf death. So really, molecular diagnosis is needed. You need to submit the sample to the lab, and we can isolate the fungus and tell you whether it's really Fusarium wilt or some other pathogen that's causing a petiole or rachis blight. In terms of, the spear leaf, it dies early in lethal yellowing, but in the Fusarium wilt, it's the last one to die. That's one symptom that can separate lethal yellowing or phytoplasma diseases from Fusarium wilt. Plus, there is no one-sided leaf death with these phytoplasma diseases. In terms of Ganoderma, you also see this die off from bottom of the canopy to the top, but it's a slow-moving disease, and the one-sided leaf death is also not observed for Ganoderma. In terms of management, as of now, there is basically no fungicide that is effective, but we have an idea that these Fusarium wilt, it is being spread from canopy to canopy through pruning tools. If pruning tools are dirty and then they carry the spores, that can help spread the disease. the diseased palm in the landscape needs to be removed and destroyed, and not to be used as mulch. These are common cultural practices that you are aware of. And in terms of Forma specialis, why that is important is if we can identify palmarum as the cause, for causing Fusarium wilt, then you should avoid planting queen palms or Washingtonias in that site. You can plant other palms which are not susceptible to Fusarium wilt. And if there is a phoenix palm that is declining and we identify canariensis as the causal agent, then you should avoid planting a phoenix palm in that site. So Fusarium wilt, there are species out there that are supposedly resistant to Fusarium wilt, and they can be substituted in term when you're replanting that site. Moving on to Ganoderma butt rot, which is caused by Ganoderma zonatum. It is a lethal disease. Ganoderma zonatum falls under a class of fungi which are known as white rot fungi, which have the ability to degrade lignin, and usually this disease usually shows up in the lower section of the trunk, and this is important when we're sampling for this disease. So Ganoderma, like I said, is a white rot fungus which can degrade lignin. Lignin is a structural component of the trunk and of xylem vessels, and it is one of the hardest compounds to break down. But these fungi, like Ganoderma, can actively break down lignin. They can destroy the xylem vessels and stop the water movement from happening. And they're not selective for lignin. They can degrade other carbohydrates as well. So the whole trunk is being rotted from inside, and the movement of food and water to the fronds is being disrupted. And that's why you see these desiccating fronds. The fungus, the wilting alone is not a diagnostic feature because other disorders can also cause wilting. So what you normally are looking for when you're looking for signs or symptoms of this Ganoderma butt rot is looking for the basidiocarp or the conch on the trunk. This is the fungal fruiting body. So if you look at this phoenix palm, you can see the bottom of the trunk is covered, and this is usually the case in the landscape. These are the bottom few feet of the trunk is covered with other, crops or plants that are grown as a hedge or as ornamentals in the landscape. So it's really sometimes really hard to see, but you see these fronds are declining. Once we cleared away, this was at the center where I work at Fort Lauderdale REC. Once we cleared away this vegetation, or when you look closely, even without clearing it, you can see these conchs. And this actually palm was completely covered with conchs, and this is fairly late in the disease. Only 1 or 2 fronds are open. Rest of the fronds are really dead. But even with so much damage, this took another year for this palm to really fall to the ground. So it's a slow-moving disease, and conchs were the primary method to make sure that the wilting that you're seeing is caused by Ganoderma and not by some other factor. And I said it was the factor because now we have an assay that we can use to find if this Ganoderma actually present in the trunk or not. But before I talk about that, even the conchs have a life cycle. You can see initially it shows up at this undifferentiated mass. It looks like any growth which is really white, and there is no telltale sign whether this is Ganoderma or not. Eventually, this undifferentiated, unorganized mass will develop into the shelf-like fruiting body and will have this white margin. This tells us that the conch is actively growing and will eventually produce spores. As the fruiting body matures and gets old, this white turns into this black. In temperate regions, these Ganoderma conchs actually fall off the tree, but in Florida, it doesn't get that cold, so they stay attached to the trunk. You can see some white. So this is the new season's growth, and the one with the black margin is the old season growth. So when you see, normally these palms are taken out of the landscape and the root stumps are left inside the ground. um, which is easier to do and less, economically burdening. But these stumps should be monitored, and as every year, as long as a stump is present and it's infected with Ganoderma, these will produce conchs, and these conchs should be removed as you see them. If left by themselves, these will produce spores, which will spread by wind and become inoculum source for starting disease in the landscape. So whenever you see a conch, you can put that in a plastic bag and throw them in the garbage. It will just rot away. It's not harmful in any way to us when we are handling it. It's just we're stopping the disease from spreading. So if the stumps have been left in the landscape, they should be monitored every year. They should be in the conchs, when they appear, they should be removed from the landscape. So in terms of, so those were the signs. Like I said, a physical presence of the fungus is the sign of the disease, and the symptoms are visible when you cut the trunk. As you go up in the numbers, as you go from 1 to 7, you're going away from the soil line. You can see this is more darkly colored as opposed to lightly colored trunk. And you can see sections 4, 5, 6, you can see a clear zone of rot. So this, it looks like if you put these sections together, it looks like a traffic cone. There is more rot at the base. As you go up, there is less rot. But eventually, when the bottom section is completely covered in rot, the palm is dead. It will take another, few months to a year before the palm actually falls to the ground. But it is a hazard. If you know the palm is infected with Ganoderma, it's dead, it should be removed, especially from areas which have high pedestrian traffic close to sidewalks or the roads. So like I said, conchs were the one way to make sure that the palm that is declining or showing desiccation of fronds is being caused by Ganoderma zonatum. But since then, since I started, we have developed this assay where we can detect Ganoderma zonatum months before the conch is actually formed. So if you have a palm that is declining in the landscape, you see this desiccation of fronds and you're not sure, and the conchs are not visible, you can send it to the lab. What I'm showing here is basically 6 panels. This is a subset of panels. There are 15 known. These all represent different species. And the Ganoderma is in this panel 3 here. And this is a subset of 15 known species of Ganoderma. So what you see, these 3 bands, which are present in lanes 1, 2, and 3, only showing up in zonatum and absent in all the other species that we tested, the 15 species. So it's really specific to zonatum. And Ganoderma zonatum is the only Ganoderma species that infect palms. The Ganoderma that comes on palms will not infect any other vegetation or any other crop or any other tree in the landscape. And if you see Ganoderma on oaks or other trees, they won't infect palms. So they have a very specific relationship, and we can, detect Ganoderma zonatum from the sawdust. There is a EDIS document out there that we have put out for how to collect the samples. It's very similar to phytoplasma, collecting samples for phytoplasma. The only caveat being closer to the soil line. Ganoderma usually infects the roots. It's a soil-borne pathogen. It comes in through the roots and moves up into the canopy, or not into the trunk, sorry. It's really 99% of the times it's base of the trunk where the Ganoderma is present. So once you sample, we suggest you sample at least 2 sites on the trunk. If you're sampling on the north side, sample from the south side as well. You can pool the sample and send it to the lab at Davie. There is a sample submission form available online on the FLREC website for palm fungal sample submission form, not to be confused with the phytoplasma sample submission form. So there's a separate sample submission form, and you can fill that out and send it along with the sample, and we can test it and tell you whether Ganoderma is actually present or is the cause for the decline or not. In terms of management, I've been working on this disease for about 4 years. The slide here is based on work from a predecessor, Dr. Monica Elliott, who worked on this for 20 years. In her experience, we could not correlate any environmental or landscape management practice that, with the presence or the absence of disease. So whether the palms are transplanted, whether they're healthy or weak, whether there's water present, or whether they're in well-drained settings, whether there's wounding, soil type, there was no relationship. And all known palm species in the landscape are susceptible to Ganoderma. We haven't seen any species that has been spared or being known to be resistant to Ganoderma zonatum. Again, there are no cultural chemical controls. We're working on fungicide solutions for Ganoderma and looking at soil treatment as well as root treatments. That is work in progress. We have done some in vitro studies, and there are some fungicides that appear to be functional. Greenhouse studies and landscape studies are pending. Again, some common methods to stop the spread of Ganoderma is not to use the infected part of the trunk for mulch. Conch should be removed as they appear, and avoid replanting another palm, just because we don't know how long the Ganoderma inoculum can be present or survive in the soil. But we are working on that aspect, and hopefully we'll have an answer for you soon. There is a third disease which is lethal, and this also causes trunk rot, but it's really different from how Ganoderma shows up. It's caused by Thiliviopsis. Thiliviopsis is also a soil-borne pathogen. It also rots the trunk, but usually as compared to Ganoderma, which shows up in the bottom of the trunk, that usually shows up in the upper part of the trunk. And the trunk collapse is the most prominent symptom. And by the time the symptoms appear, it's really too late. So normally the palm will look healthy, and eventually, the trunk will collapse. So since there are no telltale signs, you really can't predict when Thiliviopsis is going to, cause trunk collapse. Only in coconuts there is, stem bleeding. I'll talk about that in a little bit. So, like I said, as opposed to Ganoderma, which shows up on the bottom, this Thiliviopsis, it shows up in the upper third of the trunk and causes one-sided rot. You can see part of the trunk is rotted in the black as compared to this light colored tissue. And so the structural integrity of the trunk is compromised. This pathogen cannot destroy the xylem vessels, and it cannot degrade lignin, which Ganoderma can do. So the xylem vessels, there is no disruption of water movement or food movement. So the palm stays healthy or looks healthy and green. But the other tissue that holds these, vascular bundles together, that is being degraded. So the structural integrity is compromised. And depending on where the rot is happening, either the trunk will collapse, or if the crown is too heavy, the whole crown can collapse and fall down. And which again it could be problem or a risk to pedestrian traffic, or to depending on where the palm is installed. If it's high pedestrian traffic or close to driveways, that can be a concern to property damage, as well as a threat to human life as well. In coconuts, stem bleeding can be seen in some cases. There's reddish brown stain that runs down from the point of infection. It's a soft rot as opposed to Ganoderma, which is a hard rot. So if you try to push your finger through, you can actually go past the pseudobark if there is enough rot close to the infection point. So this is a soft rot, and you sometimes can see stem bleeding in coconut palms. Not so much visible in other palm types or other palm species. In terms of management, Thiliviopsis, based on our lab studies, is not an aggressive pathogen. It needs wounds to enter. We have done studies in the lab. If we don't wound the tissue, Thiliviopsis cannot cause disease. But wounds on palms can occur by several methods. There could be natural wounds like trunk cracks. There could be damage by insects or mammals. There could be disease, wounds caused by nails or during transport, or, especially during pruning. When we over prune, we're pruning green petioles. We're creating wound sites where Thiliviopsis spores can get in and cause rot. And that's one of the reasons it shows up in the upper part of the trunk and not in the bottom part of the trunk. It enters through pruning wounds or other wounds, close to the canopy. So and due to lack of any visible symptoms, there are no proven strategies to prevent this disease. And like always, try to remove the diseased palm to reduce the inoculum load and not to recycle a palm in the landscape. But wounding is a critical component of this disease to happen. That is where the disease cycle aspect comes in. We know if there is no wounding, we wounded the leaves, this pathogen can cause disease. If you don't wound the leaves, this pathogen just sits there and is unable to cause disease, which is, one aspect of disease cycle that we know now. And if we can avoid causing wounds, unnecessary wounds, we can reduce the incidence of Thaliviopsis trunk rot. Another disease which is lethal is bud rot of palms. It's caused by Phytophthora palmivora. Phytophthora is an oomycete. These are water-loving molds. These are not really fungi, and they differ from fungi a lot of different characteristics. But the main characteristic is that they love fungi. They love water, and if you can manage water, if there is water management, this disease can be easily managed. But so how this disease shows up is, as the name suggests, bud rot. There is wilting and discoloration of the spear leaf and the next youngest leaf. You can see in these pictures here, and the leaves desiccate, they turn brown and die. The bud dies. Once the bud dies, there's no new fronds that will emerge. So the palm is essentially dead. And the spear leaf and the leaves closer to the spear leaf, they can be easily pulled out. And sometimes this rot is associated with foul odor. That's because other organisms come in and start causing fermentation. And older leaves, they stay healthy for months. In nurseries, since the plants are easily visible in the containers or the small palms, you can see this discoloration on the fronds. And if you can avoid overhead irrigation, you can manage this disease easily. In the landscape, you will normally see as this open topped crown where the spear leaf is missing. The other fronds look healthy, but since the spear leaf is missing, the bud is dead. These palms will eventually fade out. They are essentially dead. And bud rot usually occurs, like I said, these are water molds. They love moisture, and during rainy seasons, it's a bigger issue. In terms of diagnosis, submitting the sample to the lab is needed to determine the actual cause for the disease. In terms of smaller palms, sanitation and water management can be helpful. In terms of landscape and, again, container palms, you can apply fungicides that are specific for oomycetes, like phosphite products, propamocarb, mefenoxam. These are fungicides which can apply as bud drench to manage for Phytophthora diseases. So switching to non-lethal diseases, like I said, this classification is arbitrary. If these diseases are not managed, these will eventually kill the palm. First up is petiole or rachis blight. Like I said, this produces one-sided leaf death. Earlier, I showed you an example from a queen palm, which is a pinhead palm. Here, I'm showing you which was caused by Fusarium wilt. There we know it was Fusarium. We isolated Fusarium from there, but the symptoms are similar. One-sided leaf death in the frond here, and there is a streak running along the petiole. But unless you submit this petiole sample to the lab, it'd be hard to tell whether this is caused by Fusarium or by a petiole or rachis blight pathogen. There are a number of different fungi that can cause petiole blight. And again, like Fusarium wilt, this disease moves up through the canopy, but that movement is really slow. Palmarum, it'll take about 2 months. This is a slow-moving disease. And they only infect the petiole or the rachis, not the leaf tissue. The pathogen is actually present in the petiole. And like with Fusarium wilt, it's the destruction of the vascular tissue. The xylem vessels are being occluded, no water movement is happening, and so those leaves are basically dying for lack of water. Petiole is the tissue to be, if you want to submit tissue to the sample, it's petiole tissue that would be the ideal tissue to identify this disease or this pathogen. In terms of the other non-lethal disease, which is common in Florida, it's Pasteurellotiopsis, caused by Pasteurellotiopsis. Pasteurothiopsis are a broad group of, it's a big genera with a lot of different species. Majority of these species are endophytes. These are organisms that live in other organisms, and they do not cause disease. But some of these species are pathogenic, and they can cause leaf spots or petiole blights on Robelinis. They're usually limited to leaves, but if they're not managed, they will eventually get to the bud and cause bud rot. Especially during winter season on Robelini palms in Florida, this can be an issue. And these pathogens are also not aggressive. They enter through wounds. So, wounding on leaves or fronds can happen, during windy season, leaves rub against each other. There can be wounds that are created, and these pathogens or spores can come into the wounds and eventually spread to through the fronds and eventually get to the buds. But generally, they can be easily managed. If you can see the fronds that are dying because of Pasteurothiopsis and remove those fronds, that can reduce the disease or load or inoculum, and you can manage this disease. There are a lot of diseases that are foliar, and these are leaf spots and blights, and a lot of pathogens, over 10 different pathogens that can cause these leaf spots and blights. They're a bigger challenge on smaller palms, on seedlings and juveniles, where you need fungicide sprays to manage the inoculum load, as well as to manage or decrease the damage by these leaf spots or blights. But in larger field-grown palms, these are not a big issue. One disease that may be an issue is this disease is a leaf blight caused by Cylindrocladium, especially on Washingtonias. You can see these lesions are small water-soaked lesions. They grow with this necrotic center. It's still surrounded by water soaking. Eventually, these lesions will keep growing and developing and merging into each other and eventually take over the whole leaflet. In Washingtonias, especially in the rainy season, or in container-grown Washingtonias, which are getting overhead irrigation, this disease is a big issue. And if you can avoid overhead irrigation, you can easily manage this disease. Other leaf spots, they stay as individual leaf spots. They don't merge. They don't, take over the whole leaf. So they're easier to manage as compared to this leaf blight disease caused by Cylindrocladium. In general, and talking about diagnostics and management for leaf spots or other foliar diseases that may be non-lethal, is, if you can remove the severely infected fronds, you can reduce the inoculum. In the case of petiole blight or rachis blight, sometimes you can see these fruiting bodies, which are not visible when it's fusarium blight. You can see this, I'm not sure if you can see, but these are small black pinheads on the disease part. This actually tells you that some other fungus is causing this one-sided leaf death, and it's not fusarium. So these fungal structures may help identify or be one characterizing feature of this petiole blight. But again, we need to culture this out and see what pathogen is this. And then again, with these foliar diseases, if you can manage water for container-grown palms, it can increase the plant-to-plant distance, increase air movement, irrigate early in the morning to reduce leaf wetness. So if you irrigate early in the morning, when the sun comes up, it will the dew and the irrigation water will evaporate, and the plants will be, or the leaves will be wet for a lesser period of time, giving fungi not enough time to colonize the leaf. It will change the environment, and avoid overhead irrigation when possible, can help manage these diseases. Again, broad spectrum contact or systemic foliar fungicides, easily available in big hardware stores. They can be applied to manage these diseases or foliar diseases easily. Another foliar disease, and this is the last slide, is false smut or Gaffiola, caused by Gaffiola phoenix. It's really common on phoenix palms. And it's visible to the naked eye. You can see these black dots, cup-shaped fungal bodies, oozing these white material, and can be easily seen without magnification. It's purely cosmetic. It's primarily on date palms, and if you see the incidence of this pathogen is really high on lower leaves, you can easily remove those fronds But in Phoenix palms, potassium is a nutrient deficiency is a big issue. So if the palm is heavily infested, don't forget to fertilize the palm with the potassium fertilizer. Give it a potassium dose and remove these additional fronds to reduce the inoculum load. So that's one caveat with Phoenix palms is they have potassium deficiency, and you should take that into account and fertilize palms with potassium when you're removing fronds to reduce disease load because of false smut. This is a leftover slide from another engagement, but APS celebrated plant 2020 as plant health, as your health is your health. Oh, okay. As plant pathologists, we have an organization, which is the American Phytopathological Society. If you're interested in learning more about plant pathology, the apsnet.org, the website here, has a lot of resources in the education tab, on diseases and pathogens, on disease diagnostics and management. If you're interested, you should look that up. They produced, for the plant health, they produce a lot of infographics, talking about coffee diseases, the impact of disease can have on coffee, on chocolate, some of our favorite foods, on bananas, on oranges, talking about, reduction in orange juice production in Florida by 72% because of this one disease. You're all familiar with that. From 2007 to 2018, the impacts these diseases can have, this is, and the aspect of disease management, of trying to develop resistant varieties, of improving in pesticide usage, of integrating other approaches like biological control, is something that, as plant pathologists, we work on. But having said that, this is my final slide, and I'm open for questions. My contact info, and again, Fort Lauderdale REC website, if you type in FLREC palms, you come to the first page, and there is the palm sample submission form for fungal diseases of palms. Thank you.
If you've got any questions, feel free to put them in the chat, or go ahead and unmute yourselves, or you bring up your video even. And because we got, I don't know, it's almost 1, but probably can go a little bit over as well. I think Henry, so my fellow agent from Miami-Dade County, he's got a question here on any recommended management for Pestulopsis.
Hey, Henry. Yeah, it's because it's mostly a foliar disease. It's generally, broad spectrum fungicide that you would use for other crops. They can be used for managing foliar diseases. And if you can detect it early enough, just pruning the frond should help reduce the load. So some culture practice before you take the fungicide route. But broad spectrum, you're looking for, on the label, you're looking for the terms broad spectrum and systemic. And then you apply that as, a foliar spray according to the label rate, and you should be able to manage the disease.
We got another question here from Beth Alman. And Beth, you might need to clarify. She's asking, does stump grinding spread spores? And so I don't know if it's any specific fungal diseases or in general, but
probably Ganoderma.
Yeah, actually, in fact, I had in mind an oak tree that we need to remove in my community that has Ganoderma. Should we avoid stump grinding?
I wouldn't say avoid it, but try to manage it. The spores are produced by the fruiting body. So when it produces the shelf-like fruiting body, and if you look under the surface, there are a lot of holes in it, and that's where the spores drop from. So inside the trunk, there won't be any spores. It'll be more mycelial fragments. So yeah, I mean, it is.
Okay.
Yeah,
Thank
grinding
you.
can help spread it, but it can be managed if you lay out some try to collect that as best to your ability as possible. Yeah, but grinding.
Thank
Yeah,
you.
yeah.
Here's an interesting question from Chuck Lippy. Are Livistonia species still considered not susceptible to fusarium wilt diseases?
Hi, Chuck. So this, I personally haven't done the testing on Livistonia. The data I have is from this 2018 publication from Monica, and in her analysis, when she tested, using Fusarium oxysporum formaspecialis palmarum, which is known to be associated with queens and Washingtonians. When they tested it, or when she tested on Livistonia, she did not get any mortality. So that was considered as a resistant species. But if you have data, or if you have evidence for otherwise, I can work with you and try to figure that out. I hope that helps.
I've got kind of a broad question. I know that biologically palms are closer to grasses, and do you have any like broad type diseases that can jump from grasses or other relatives that are closer to palms, like kind of jumping from palms to other things that are a little bit biologically similar? Like
Well,
ponytail palm or some of these things that we aren't really necessarily a palm, but are still kind of in that area.
yes, yeah.
Monocots.
So yeah, I can think about several examples. One is, okay, let's start. There is this disease in strawberries, which is being caused by a Pasteuroteopsis, which is closely related to Pasteuroteopsis, is Neopasteuroteopsis. They don't know where that source is coming from. So they're looking at other surrounding vegetation, and Pasteuroteopsis affects a lot of different crops. I'm not saying there is a one-to-one correlation, but there is a possibility. Some of these pathogens which we find on palms, they have broad host range. One example is Pasteuroteopsis, okay? The others are like Ganoderma, which I said has very specific to palms, but it has been in surveys found on another monocot, bamboo. There are very few cases, but it has been isolated from bamboo. And that was Ganoderma zonatum. And this is work done by Jason Smith, who was in forestry. He has since moved. He was a professor there, and his graduate student, Andrew Lloyd. And they have an interesting study on this. A third example, there's one more pathogen I was thinking of. Oh, Thaliviopsis. Thaliviopsis is the one we are working with is Thaliviopsis ethecetica. Here I had listed Paradoxa, but Paradoxa is a broad umbrella term. There are at least 2 different species of Thaliviopsis that can infect palms, but they don't only infect palms, they can infect sugarcane. It's a big issue in sugarcane in Brazil. It's a big issue on pineapples. It was first isolated, Thaliviopsis was isolated from pineapples. It was called the pineapple pink rot disease. So Thaliviopsis has a broad host range, but mostly limited to monocots in this case, in this species case. And so, bananas, yeah, pineapples and sugarcane is our other host that Thaliviopsis ethecetica that infects palms can also go on. So some of these pathogens, one pathogen that I didn't talk about is Fusarium pluriferatum, which in California is a known concern on palms, but it's a broad host range pathogen. Infects other fruit crops, a lot of other ornamentals, and we have seen some evidence for pluriferatum in Florida as well. So we have started to monitor for that as well. But it is a broad host range pathogen. I hope that answers your questions.
Yeah, it's horrifying. I'm sure it's seeing some of the things that come in through the lab, and I mean, you see probably a lot of the worst cases in the state. So, but I mean, it is, especially like given like how much palms cost in terms of landscape management. They're often some of the most expensive things out there, and then and we see it all. But I mean, is I guess a follow-up to that, the graphiola, how have you been seeing that being particularly widespread lately at all, or is that kind of one of these ones that pops up occasionally in some years? Maybe there you have the right circumstances that it's highly present. In other years, it's not. Like, I mean, that was one that I just found out about recently. So I was curious if you've seen it a lot.
Well, yeah, we haven't received any samples for that, but usually on Phoenix in the landscape, it is present, and it's not really, a lethal pathogen, so it's easily managed. So in the lab, we haven't received any samples, but when we are out and about, we do see it on Phoenix palms frequently. Yeah.
Okay. There's a phytoplasma question, which I guess is hard to avoid too, diseases. So how widespread in our area is lethal bronzing? And I think probably the question is probably what they're referring to is like Southeast Florida. So probably most of the Tri-County area.
Yeah, I mean, I could defer to Henry and John on that, but Brian Bader is working on it. He has this whole map on which counties this has been discovered, but based on what I've heard in his talks, it's fairly widespread in Southeast Florida, and it's still spreading. It's being carried around by an insect, and basically follows where the vector moves. So yeah, in terms of percent area coverage or percent species knocked down, I don't have the exact numbers, but yeah.
So, and I guess if unless something else pops up here, I guess my last question is in terms of prevalence or things that you get mailed in terms of the lethal diseases that you covered, are any of them that you are seeing more than the others, or is there? I mean, is there anything that's occurring lately that might be like an uptick or anything like that? I know you were talking about the disease triangle, and that like there's certain times a year that things pop up, but is there like an overall trend from year to year that you're seeing certain disease becoming more prevalent?
Well, this past year, we received a lot of samples on Phoenix, which on initial assessment looked like could be Fusarium, but they were mostly, petiole or rachis blight pathogens. Like I said, they show up as similar symptoms, and there is eventually you keep pruning these dead fronds, and then the canopy size keeps getting smaller. But these can be managed with bud drench, or I've been working with one HOA. So far, the palm has survived for 2 years. The canopy has not bounced back, but it's still surviving. So, we had more cases of what looked like Fusarium wilt turned out to be petiole blight, especially in phoenixes. And it is a slow-moving disease, and hopefully can be managed. The other was something that Romina, who runs the tropical REC, she identified on coconuts, was Lasiodiplodia, which is causing petiole or rachis blight, which might be something we should be looking at. And we recently did some pathogenicity assays, and Lasiodiplodia may become a problem on coconuts in the coming years. But yeah, something to keep your eyes on. The Fusarium proliferatum that I mentioned, in California, they call it as the symptoms that appear on the leaves are what they label as disfiguring blight. Basically, the frond appears to be chewed up. The frond normally has the leaflets are tapering as you go towards the tip. But in this case, the leaflets appear as they have been chewed up by an insect. So I should have included some pictures. Hopefully in the next talk I will. But if you see, Phoenix palms like Robelinis with disfigured fronds, maybe Fusarium proliferatum is could be the issue.
Okay.
So yeah, there are things, or there are diseases on the horizon that, you know, may become issues in the next few years, but yeah, we'll keep our eyes and ears open.
Okay. I went ahead and launched the quiz too for anybody that's still here. If you could go ahead and fill this out. This helps us be able to evaluate what you all learned. And then I guess maybe like in the next, if couple minutes while you're filling that out, if you've still got a question, like go ahead and throw it out there. So I see Steve Corbet.
Yeah, just a quick question. Is there anything that I should be aware of in northern Florida? I'm way up North Florida that isn't as prevalent as down in like West Palm area. Because we do actually get a winter. We had snow this year. I'm Florida, but we still got 7 inches of snow.
Yeah, I heard about that in North Florida, REC. Yeah. Yeah. In the north, I think, you know, like I said, some of the, like the Pachyloteuthis diseases on Robelinis, but I don't know how many, you know, the diversity of palm also decreases as you go up north. You're not getting coconuts, you're getting more sables, maybe a few phoenixes or pindo palms. So yeah, phoenixes, it's petiole blight or fusarium, that'd be an issue in phoenixes. But yeah, I would like to talk to you more and see, you know, what you normally see up there. But yeah.
Yeah, we get a lot of bronzing more than anything else.
Yes, yeah.
That seems to be the most common.
Yes, yeah, I got you. Yeah, fungal diseases, yeah, it's more bronzing as compared to fungal.
If I'm relegated to watching this on an iPhone, where can I get the quiz? Can you email that?
I mean, we can. The quiz is mostly for us, like for us to be able to kind of evaluate what you all have learned. But I will follow up, and if you can, the thing that I will give everyone at the end is a link to the recording, so that you'll have that to go back to. But the quiz really is, it's more for like our side to show that like we're actually getting you to learn something through the webinar.
Yeah, I did pay for the ticket. Somebody just commented on that. So anyway.
Oh yeah. No, if you need CEUs, like I'll be processing these afterwards here. So everything gets I send all the information to FNGLA, LIAF, ISA, and FDAX. So okay, so what causes multiple unopened spear leaves on Phoenix species?
I'm not sure on that. I know there are multiple unopened spears on Bismarckias. Phoenixes, it's not that common, but I do have seen that, but I'm not sure on what might be the cause. If anybody else has any, you know, if you have seen this before. Oh,
Yeah,
Chuck
yeah,
says it's boron deficiency.
That
Thanks,
sounded
Chuck.
more, yeah, thanks, Chuck. That sounded more like a deficiency.
Usually boron deficiency is also associated with leaf curling and disfiguring, but maybe early stages it's unopened fronds. But in Bismarckias, you have multiple unopened fronds.
I've seen it on Bismarckias frequently, but now I'm seeing it on Phoenix sylvestris that we're getting multiple ones, like 5 at a time, and they're all unopened.
Yeah, so David, what I'm trying to say is in Bismarckias, it is a physiological feature. It's probably not a deficiency. But in Phoenixes, it might be a deficiency, is what I'm trying to get at.
Thank you.
All right, well, I went ahead and ended the poll. I want to thank Dr. Dhillon again for being here and covering all these different and somewhat horrifying palm diseases. Yeah, if you need identification for any of these fungal diseases, like make sure to check out his website. And he can evaluate them like in a laboratory and give you results for it as well. So thank you again, Dr. Dhillon, and
Thank you.
look forward to seeing everybody next month, hopefully. Take care.
Yeah, thank you.